Dr. Irwin also serves as a member of the Executive Committee for Neuroblastoma. She is also on the Executive Committee of INGR, the International Neuroblastoma Research Group, and the NANT Scientific Review Committee. She has led the Children's Oncology Group efforts to develop and modify risk classifiers in neuroblastoma that incorporate the INRGSS with clinical and biologic prognostic factors, including segmental chromosomal aberrations, that led her to her most recent seminal publication in the Journal of Clinical Oncology that was published last month. This publication really highlights Dr. Irwin's in-depth understanding of biology of neuroblastoma, coupled with her clinical expertise and skill in harmonizing international collaborations. Thank you for joining me and us in welcoming Dr. Irwin for her seminar, Identification of Biomarkers, Models, and Targets to Improve Outcomes for Precision Approaches for Neuroblastoma.
Thank you so much, Beth, for that very kind introduction. It's a real pleasure to give this talk. I was going to say at St. Jude, but virtually at St. Jude, and to update you on neuroblastoma. And I'll be pleased to take any questions at the end or in the extended Q&A because I am going to go over quite a few different topics today. I have no really relevant disclosures for today's talk. So today I'd like to talk a little bit about some of the work that I've done together really with a group of people, translational, clinical, locally and internationally, to help build a neuroblastoma program at SickKids, which was really started in 2008, several years after I joined the faculty. And the three objectives that I would like to cover today in a bit of a whirlwind are, as Beth mentioned, there's a risk classification update, and I want to start by talking a bit about that and why we use risk classification in neuroblastoma. I also would like to tell you a little bit about some of the work that we've done identifying genes and pathways to inform the treatment for metastatic neuroblastoma, and then end on some exciting newer work we're doing in single-cell RNA sequencing to really try to dissect further metastatic potential and other lessons about possible future biomarkers for neuroblastoma. So I think most of you know what neuroblastoma is, but just a brief introduction that it is a neural crest tumor that originates from the neural crest tissue. It can occur anywhere along the sympathetic nervous system, or more commonly in the adrenal gland. And the average age is 17 to 18 months. Although it represents only about 10% of childhood cancers, still, unfortunately, it represents a higher percentage, about 15% of cancer-related deaths, usually due to metastatic disease. The thing that has attracted me to neuroblastoma and many is really the heterogeneous presentation of its biology and the mystery really of the fact that very young children can have diffuse metastatic disease that can spontaneously regress, whereas older children often have much more metastatic and resistant disease. And so the areas I've chosen to work on that I'll talk a bit about today are really trying to identify more biomarkers, develop some preclinical therapeutic models, and I'll show you some data for maybe some new exciting therapies, as well as looking at pathways and not just single genes that might be involved in determining this difficult metastatic spread problem. So this slide I created really just to highlight always the diffuse spectrum really of what we see in neuroblastoma. And then I hope to show you how that really is reflected in the biology. On the left side, you can see the most easily treated or not even needed to be treated neuroblastoma that often are small masses, even detected prenatally, or metastatic disease in newborns that can spontaneously resolve or differentiate. And we often just observe or occasionally do surgery. In the middle are the group of children who are often young with regional tumors, such as the one shown on that chest CT, who often need minimal amounts of chemotherapy and maybe surgery. And then on the right are the 50% of children that really occupy most of our time, which are the highly resistant metastatic patients that still require multimodal treatment. And what we know is this heterogeneity is really reflected in numerous biological factors as well as strong prognostic factors, some of which are genetic. And shown on this slide is the robust data we have from international work as well as the data we collect in the Children's Oncology Group, showing our four most strong prognostic predictors. So age and stage on the top are, continue to be prognostic no matter how much sequencing and other important biomarker work that we do. But on the bottom are two other factors that we know historically and still today continue to be robust markers of prognosis as independent factors: the amplification of the MYC oncogene and histology. This slide is to remind me to let you know about a really nice paper that I was involved in together with Sarah Federico at St. Jude's and Sue Cohn and others, where we went through the history of risk classification in neuroblastoma, which really dates back to the 1970s when Audrey Evans at CHOP started to recognize the prognostic strength of things like age and stage and various other sort of clinical factors, which over time on this timeline, you can see have started to involve things like MYC status and increasingly more and more markers. On the right is a figure from the paper just showing how the classifiers over the years from the 1990s to 2021 have continued to include several factors like age, but have started to add different factors. And increasingly, we're hoping some of the bottom rows, which are more genetic markers and pathway markers, will start to be used and are currently being looked at in a prospective manner. And what we've been using for the last almost 2 decades in the COG has been this risk classifier shown on the bottom. On the top part of the classifier with the blue box, you can see the factors that we've been using over the last 2 decades, the same factors we've been talking about. And on the right-hand side are the risk groups. So on this slide is the results of the last 10 years, really, from 2006 to 2016, with 5 years follow-up from the paper that Beth referenced that just came out in the Journal of Clinical Oncology, showing our most recent outcome data for patients that we've classified on that classifier I showed you as low, intermediate, or high risk. And as you can see, the survival for low and intermediate risk is excellent, and for high risk, we still have work to do. And the types of treatments that we used for these patients during this 10-year period are shown in the boxes on the right and mirror that first slide I showed you of sort of very low intensity types of therapies versus intense multimodal therapy for the high-risk patients. And I'll just make a brief note now because I won't speak that much about our clinical treatments, but happy to address that later. But to say that for low and intermediate risk patients, really our goal in the Children's Oncology Group and internationally is to continue to reduce therapy as informed by the biology and other factors about each patient. Whereas for high risk, we haven't really been able to back off, but we are hoping to be able to figure out which patients really need all this therapy, which patients maybe need more, and which patients can use less. And then the work that was summarized in the Journal of Clinical Oncology paper was really brought to our attention, and the need for it, the rationale for it, was to take our risk classifier shown on the left from the Children's Oncology Group and really try to harmonize that and move forward to align with the international or INRG classifier, which is also used by our European colleagues. And these two classifiers use different staging systems and somewhat differently account for some of the biological and clinical risk factors. So our goal in analyzing our data from the last decade in the Children's Oncology Group was to really harmonize these two risk classifiers to look at our patients who were treated more recently than many of the former cohorts that have been published were treated, to change the way we stage patients and to be able to look at this new staging system, the INRG, and to incorporate new biomarkers like SCAs or segmental chromosome aberrations. And importantly, to do this all in the context of the modern therapy, which we know has changed significantly, especially for high-risk patients. And I'll make a small note that as we've been working on this second risk classifier, which I'll show you in a minute, we're also starting to get poised and do studies to start incorporating more genomic markers into our next risk classifier down the road, because this takes so long to plan these types of studies. And so to come up with this new classifier, which will now moving forward be the classifier we use in the Children's Oncology Group, we looked at the EFS and OS, or event-free and overall survival, for various combinations of clinical and biological prognostic factors in our 5,000 patient cohort. And shown on the next slide is the chart, which is in this publication, which I'll refer you to, that summarizes our new risk classifier. And what we wanted to do was really show the algorithm or the strategy in a chart rather than the complicated figures we've done before, to show you how you can take a patient here with locoregional disease, either L1, which has no image-defined risk factors and is easily resectable, or L2, which has imaging-defined risk factors and often are large unresectable tumors, and then follow along to see how the different factors lead to different risk groups based on the event-free and overall survival that we analyzed in the publication. And on the right is just summarized the metastatic disease and how we do this for patients who present with metastasis. Whereas you can see on the right side of this graph, it's almost impossible to not be high risk or intermediate risk once a patient does present with metastatic disease. And I'd be happy again to answer any questions in the period at the end about these new classifications. On this next slide just shows our outcome using our new risk classifier, and you might see that it doesn't actually look all that different, but what is important to recognize is using what we believe to be a more optimized classifier that's harmonized with our international colleagues, we believe we're better able to put our patients into the various categories or buckets to determine the optimal treatment based on all of these different risk factors that they have. But for the rest of the talk, what I'd really like to talk to you about is some of the insights that we have or are trying to have for this high-risk group to move forward to really improve their prognosis. And just to summarize for the group, the patients we're talking about, generally, at least half of them, are patients who present with stage M or distant metastatic disease and are over 18 months, regardless of their biology. Or patients who are infants and toddlers under 18 months who have some sort of unfavorable biomarker, and then a smaller group of locoregional non-metastatic patients who have very unfavorable biology, such as amplification of the MYC and oncogene. And this group makes up about half of the patients who we all treat with neuroblastoma, but the patients we, again, disproportionately spend our time and resources taking care of. This slide provided by Arlene Naranjo, our statistician for the Neuroblastoma Committee, really represents Kaplan-Meier's over every 5-year period. And I hope you can see that whereas we haven't gotten as high as we'd like or as high as the leukemia groups have gone, we have actually made significant progress over the decades as we've improved therapy and included different types of therapy for this subset of high-risk neuroblastoma patients. And to remind everyone, the current therapy, which I often describe to families and patients and trainees as a triathlon, includes very intensive chemotherapy for about 6 months, often including a surgical resection during that time, 1 or more autologous stem cell transplants, followed by post-stem cell transplant therapy that includes immunotherapy with anti-GD2 antibody as well. And even with this full triathlon of therapy, you can see by the purple Kaplan-Meier, we're still in approximately the 50th percentile for event-free survival, although we are starting to see some improvements. But unfortunately, 50% of patients then do recur. And although I won't speak about the current standard of care for relapse patients, shown in the red box is just a summary of really the go-to therapies that we now use, which have improved over the last several years for patients with recurrence, which when I was a trainee was pretty much completely fatal within months or maybe a year of presentation of recurrence. So chemoimmunotherapy has approximately a 50% response, and that was shown in a study from the Children's Oncology Group that's referenced here. High-dose MIBG therapy leads to a response in about 1/3 of patients with recurrent disease. And then, of course, there's chemotherapies. And for subsets of our patients, increasingly, we are able to identify targeted therapies. But overall, complete cure and overall survival remains poor for these patients. So something I've been very interested both in my children's oncology role and in the lab has been identifying biomarkers as well as targets and therapies and really elucidating why this happens and what makes what is responsible for the patients who represent the green arrow and are going to do better and maybe don't need all of this therapy, and those that are going to have a poor outcome despite this triathlon of therapy. And so the way our group and many others has addressed this in the last 5 to 6 years has been really taking advantage of the explosion of next generation sequencing and other genomic technologies. And I've summarized many, many papers and thousands of genomes and exomes on neuroblastoma cases here to say that whole genome and whole exomes have really taught us that there's, just like neuroblastoma's clinical presentation of heterogeneity, there are no real recurrent somatic driver mutations, and instead it's quite a spectrum. The most common gene that's mutated is the anaplastic lymphoma kinase, or ALK gene, in about 10% of cases. And then I've listed many of the other genes that have been found to be mutated, although at a lower incidence than ALK. And I've just highlighted with asterisks the telomerase maintenance mechanisms, which is an interest of mine and us in the COG. And we're looking at this very closely to see if this can really be a biomarker in our next risk classifier, and also potentially a clue to interesting therapies for patients where these mechanisms have gone off. But what we do know from neuroblastoma as well as other cancers is that the mutations that we see at diagnosis are more common, and often even there's clonal evolution and new mutations that arise at relapse. And we certainly weren't the first, but we've done a large scale sequencing project at SickKids, and actually also part of a large Canadian initiative called Profile, which involves all of the pediatric sites across Canada. And in this study, we've been doing genomes. So we've been doing exomes, deep exomes with a custom gene panel, RNA sequencing, and whole genome sequencing on patients with hard to treat cancers or recurrences. And I'm just going to summarize the neuroblastoma data, which again really reflects what's been seen in other centers. We do use our data clinically. Most of the panel results can be turned around within weeks. So that is clinically usable often before we have some of the other very interesting, but often less clinically relevant results. We had 42 neuroblastoma patients, which represented a quarter of the solid tumor patients and about 14% of all of our patients, and all diagnoses are essentially potentially eligible. And I've just summarized some of the data from neuroblastoma from one of the figures from the submitted paper, which is for all of our tumors, but essentially for neuroblastoma, about half of our patients had something actionable, either in the germline or the tumor, and that included, of course, as expected, 7 ALK pathogenic clonal variants and aberrations in the RAS-MAP kinase pathway. Another area of interest, especially because this program is really led by both Anita Villani and Adam Schlein, who leads our clinical genomics cancer program, has been looking carefully at the germline and somatic landscape of DNA repair and homologous recombination genes, as well as the presence of a DNA signature that signals potential HR deficiency and DNA repair deficiency in tumor cells. And from this work, we've been very excited to come up with some strategies initially used in a single patient study that we have here, and now being expanded and hoping to collaborate with the St. Jude group on the ONET study to be looking at PARP inhibitor strategies for neuroblastoma and other solid tumor patients that maybe have some clue moving forward of some aberrations in this pathway. And I just want to mention that we are also, like many, including I'm sure many in the audience, interested in circulating tumor DNA, and those studies are underway for a subset of these patients. As was mentioned, and I just want to highlight, ALK is really an important area for neuroblastoma, and as we move forward, a very important therapeutic option for our patients, both at diagnosis and increasingly at relapse, because it's found in about 10% of patients and actually a higher number at recurrence. Just to mention, it's also found in about 1% of neuroblastoma patients in their germline to be mutated, and it's usually in the kinase domain, shown in yellow on the right-hand side. It's now being used in our upfront COG ANBL 1531 high-risk study for those who have an aberration detected at diagnosis, either a mutation or amplification. And many of us are using it now for patients when we find these clonal and maybe even subclonal variants when they recur. Shown on the left is a CT scan from one of my patients who failed all therapies, and was one of the first patients we sequenced on KICS, found to have a hotspot activating mutation, and was enrolled in the Novartis ceritinib trial. And 6 months later, you can see had an excellent response and was able to remain on treatment for 5 years. I'm disappointed to tell you that Unexpectedly, the patient has had a progression after complete stability for over 5 years, and we're currently sequencing the recurrent tumors to see if there's a clue in their genomes to explain this sudden resistance and metastatic spread. And for the next part of the talk, I want to talk about thinking not just about single genetic alterations as targets, but instead pathways themselves. And so we became very interested in the RAS-MAP kinase pathway because of some other interests in the lab, and also because several groups, including the Maris lab and several European groups, had shown that this pathway is upregulated potentially more selectively at the time of recurrence. And this is the work that I'm sharing of a very talented a PhD student and now a postdoc in the lab who wanted to interrogate the role of sort of a RAS regulator. So SHP2 is encoded by the gene PTPN11, and SHP2 itself is a tyrosine phosphatase that we know regulates activation of the RAS pathway. And we've been looking at this in other cancers in collaboration with some other groups. And Yvette wanted to ask if SHP2 inhibitors might somehow be specifically important in some of the neuroblastomas, either with or without RAS-MAP kinase signaling abnormalities. And so we got a hold of both publicly available and other inhibitors, including 3 allosteric SHP2 inhibitors shown in the middle panel. And that tested several of our neuroblastoma cell lines in the lab, of which we knew the status of the RAS pathway. And on the right-hand side, the blue cell lines essentially are those that have known RAS mutations. And you can see that using the tool compound SHP099, a Novartis compound, those were the most resistant lines, which wasn't actually necessarily what we had predicted, but is clearly what we've seen over and over again in extended panels and summarized on the right side to show you that RAS mutant lines appeared to be actually more resistant, at least to the current SHP2 inhibitors that are available. We then set out to figure out if we could think of a strategy that would work in the blue cell lines or the more resistant cell lines that maybe bypassed SHP or collaborated somehow with SHP inhibition. And so in this slide, I'm just summarizing a lot of work, which again is published, and I'd be happy to talk more about at the end, where Yvette looked at other commercially available inhibitors, some of which are being studied in children, that target the RAS-MAPK pathway downstream of RAS and SHP2, including BRAF, MEK, and ERK inhibitors. And specifically in the panels I'm showing you on the right, we focused our initial studies on combinations with MEK inhibitors because we knew there was work going on in lung cancer and thought that might be the easiest to translate to the clinic down the road. So that combined in animal models and lots of cell data I'm not showing you, SHP099 and trametinib as single agents shown in the middle 2 columns of the panel. And then in combination, you can see there was the best effect when we used those 2 drugs together in short-term experiments, which were somewhat time limited due to the pandemic when most of this work was started and completed. And on the bottom are the similar survival curves. So we actually believe there's promise in this combination. And what's exciting is it's not just neuroblastoma, but we've had discussions with groups studying nerve sheath tumors and other difficult to treat solid tumors of childhood, and are very interested in thinking of moving these forward and are working with Novartis on the more clinical grade drugs of SHP099, such as TNO155, that potentially have better bioavailability and may even have some single agent activity moving forward. But what we've spent a lot of time in the lab doing is really trying to elucidate more of the mechanistic pathways that are, and not only taking, vulnerable pathways that have been already discovered, but trying to find new ones, especially given that recurrent driver mutations are really rare, as I showed you. And there's this heterogeneous spectrum. So, we can only get so far with every genetically engineered model as well. So we set out to figure out if we could identify pathways governing metastasis. And so the work I'm going to tell you about is a combination of published and unpublished work, really taking advantage of a model we've tried to grow in the lab to try to understand and model metastatic disease. And shown in the pictures are just some of the sites that most of you who are clinicians in the audience know to be common sites, especially the bone and bone marrow. And on the right, the CNS, which is becoming an increasing problem as we get better and better at treating neuroblastoma metastatic disease at diagnosis. We have spent a fair amount of time working with Daniel Morgenstern when he was initially a fellow at Sick Kids and now on faculty, trying to understand the implications of the pattern of clinical metastatic disease. And I'll refer you to those publications for some of that exciting work done with the INRG group. But really, I'll focus on this talk on some of the biology and what we've figured out using these models and are still trying to work on now in terms of understanding heterogeneity using single cell sequencing. So there was a student who I'm showing with the arrow, Alex Song, who got his PhD in my lab and was working closely with David Kaplan's lab and Kelly Fathers, who's shown on the left, who was a postdoc, and now Yagnesh Ladmar, who's taken on a lot of this from Alex, who's now a postdoc in Kip Stegmeyer's lab at the Dana-Farber. And what we wanted to do is make a mouse that basically looked like a patient. We wanted a mouse that would develop bone disease, potentially brain disease, and could be used for a variety of different focuses in the lab. And so there's many models, many of which we still use in the lab and will continue to use for modeling neuroblastoma metastasis. But I think what I wanted to highlight is that each of these models, just like the model I'm going to tell you about, have their own caveats and don't necessarily lead to the type of model that we were looking for, at least for the initial studies that we wanted to carry out. And in particular, I'll just summarize some of the caveats and our goals. We're trying to identify neuroblastoma metastatic models that had the frequency and severity of bone metastases that we see in our patients. And most of the models I outlined on the previous slide really don't get large osteolytic bone lesions like our patients do. Very few have shown any brain metastases. And there's a significant inability in a lot of the models to image and quantitate over time metastatic disease, although much progress has been made in that area since we started these studies over time. So the model we started working on about 8 or 9 years ago now was a model really pioneered by the Massaguet Lab at Sloan Kettering, where intracardiac injection is performed and in vivo you select metastatic subpopulations. So shown in this schema is a mouse that has labeled neuroblastoma cells. For our initial experiments, these were done with an established line, SKNAS, that were labeled to be able to image them and also select for them. And those cells are injected into the heart, and then we allow them to move through the circulation and form metastases in the mice. We image them multiple times, and then we're able to take out the metastatic tumors, and in this case, the metastatic limb, as shown in this X-ray, with superimposed bioluminescence. We take out those tumors, culture the cell, select the cells, culture them for a very short period, re-inject them, and do the same process over and over with the goal of developing increasingly metastatic subpopulations. And these are just a few pictures to show our mice that develop these metastases. The large colorful area in the middle is really the honing to the adrenal gland, which is not unexpected for neuroblastoma. And then there are multiple areas you can see. Hopefully you can appreciate the limb metastases as well as skull and brain. And we wanted to quantitate this. As you can see on this chart, we wanted to look to see by bioluminescence if the progressively more selected cells had more signal and were thus more metastatic. And you should be able to see that B1 and B5, which are secondary and tertiary collections of these cells, were more metastatic and had a higher burden of disease measured. And these are just CTs of the bone to show you again from control all the way to the more recently collected subpopulations. There are lytic lesions shown with the arrows that stain for TRAP, which shows you the activated osteoclast. And then these are brain lesions in the mice. Some of them are dural and some are parenchymal. And so this is just a schema to show you the types of cells that we've developed that I'll show you in the next few slides, what their characteristics are. So the parental cells are the original ones, or P, and then the B1s are the common ones that go to bone a bit more than brain in the first selected group, and then B5 and BR2. And though I'll focus on these, much of the informatics that we've done and many of the validations are done with many other cell lines as well. So in order to prove the utility of these, and in order to be able to do some studies in vitro, which is much more scalable, we used very simple Boyden chamber assays where we look to see how invasive and migratory our cells are, moving from an upper chamber to a lower chamber shown in orange. And so on the lower left-hand corner, you can hopefully appreciate the B1 and the B5 subpopulations are much darker, meaning more cells have gone through and have migrated and invaded, and that's quantified on the right. And then we wanted to use these various cell lines, which we knew in vitro, and I showed you a little bit of our data in vivo showing they also migrated and metastasized in vivo more, to see if we could identify differences in expression that maybe could be a marker of what's happening in our patients. And so to try to understand the genetic differences, we at the time were doing microarrays. It was quite a while ago, and this was done on 15 of our subpopulations. And we did semi-supervised clustering of the microarray data and developed 2 major clusters. And you can see the parental cells clustered quite differently from the metastatic subpopulations. And doing this, we were able to use the top differentially expressed genes to develop metastatic and parental signatures that we were able to leverage in some of our other experiments. But in order to determine if this really reflected what I told you was our goal, that this model actually looks like our patients in some way, we wanted to ask whether these genes that were more metastatic or more parental in the mouse actually reflected something about our patients. And so we leveraged a lot of in silico data to understand if our signature, here it was our 75 gene signature, could actually predict outcome. And what Robin Hallett, a bioinformatics specialist in the lab, together with Alex, showed was that actually the metastatic signature did at least seem to correlate with patients who had an overall poor prognosis. This was in all comers of neuroblastoma, and we also saw a similar effect, albeit less robust because of a smaller data set in high-risk patients as well. And so the question in the lab is, can we manipulate the genes or find drugs that can kind of reverse these signatures in patients? Well, in mice first, and in cells, and then in patients ultimately. And so on the next slide, before I get to the unpublished data, I'll just summarize a lot of data, some of which is published now, just showing the validation of the model. So what we did for several different genes that were upregulated in our metastatic cells and metastatic tumors was to basically turn them off by using shRNA or CRISPR, or for the 2 with asterisks, and for several others since, pharmacologic inhibitors, and ask whether we could rescue the metastatic phenotype. So by looking at invasion assays in the cells or in vitro, or metastatic spread in vivo. And with the ones listed here, we were able to see rescue. And importantly, I want to point out that some of these, again, were inhibitors, as shown in the small inset diagram there, where the mice treated with the SPHK1 inhibitors or sphingosine kinase inhibitor, SKI inhibitors, you can see that it did block metastasis in vivo in our metastatic model. And we also have looked at inhibitors of the YAPTAZ pathway, which are much earlier in development, but we know this pathway is upregulated in recurrent neuroblastoma as well as many other pediatric tumors. And on the other side, we tried to look at genes that were downregulated in our metastatic tumors in the mice and in patients and ask whether if we overexpressed, we could rescue. And that was also the case. But of course, this represents a much trickier sort of therapeutic approach moving forward. And more recently, we've started to use this model to interrogate genes where we're not sure if they have a role in metastasis, but have some exciting, compelling data to suggest it might be the case. And in collaboration with Ronnie George, and actually very recently published with Hao Huang in her lab and Alex Gaunt in my lab, we wanted to look at a new sort of functional role for ALK, or the ALK gene that I've already talked about, in mediating metastasis. We know a lot about ALK in terms of promoting growth, but Ronnie and her group had identified a cleavage site in the ALK protein shown in this schematic, where if you block the cleavage by introducing a mutation, you actually get reduced migration promoted by the ALK protein itself. And so our question, shown in red, was really, does this extracellular domain cleavage that mediates migration in vitro also affect metastatic spread in vivo? Because this is an under or non-appreciated potential role for ALK. And so what you can see here in panel A and B is that what we were able to see using actually both a tail vein injection model as well as our intracardiac model was that when we introduced a CRISPR variant of ALK that was unable to be cleaved, shown on the right, those cells in vitro, just like in vivo, were unable to promote metastasis And you can see on the bottom, we quantitated this, so it was not just fewer metastasis, but a smaller burden of them and increased survival for these mice. And this was done in a few different cell line backgrounds, and also, as mentioned, using 2 different models. So we think there is a utility as we all identify new variants, also to not just thinking about their role in growth promotion, but also in metastasis. And this may be one way of doing it, where we can actually also potentially look at drugs in a in vivo system. And so for the final part of the talk, what I want to show you is how we can maybe use this model to start to really start understanding the heterogeneity of neuroblastoma. And of course, this is a very active area in many tumors, and I'm only going to speak about it in this model and for neuroblastoma. There's, of course, many possibilities, but there's probably 2 main ways that we think about this. The first is that all cells in a tumor have the potential to metastasize, versus the second possibility shown on the bottom, where maybe only a subset, like the purple cells or the needle in the haystack, or those with stem cell or other promoting properties that can go on to metastasize to the bone in the brain. And this is work from Alex Gaunt, a postdoctoral fellow, and Alice Shan, who is a PhD student in the lab. And what they wanted to ask using single cell sequencing now is, could we find this subpopulation of purple cells with increased metastatic potential to the bone and or brain that we weren't able to detect with our bulk sequencing techniques? And really, ideally, what we were looking for as a second step was a marker that was not only detectable, but that could be selectable, or we could use to tag and pick out those cells from the needle in the haystack. And so I think many on the call know about our single cell RNA sequencing, but essentially a way of taking out each cell and individually barcoding and sequencing that. And on the right side are plots where we are able to show in these dimensionality plots the transcriptome of each single cell shown as a dot. And then they're clustered in groups shown here or represented by different colors where they cluster together because of the similarity in each cell's transcriptome. So here there's 6 clustered in this diagram, and on some of the next 5 or 6 slides I'll show you, you can see how we've tried to do this within the context of our metastatic model. And so the first question we wanted to ask was, is there heterogeneity between those parental, those initial cells we used, and the cells that went on to metastasize? So again, could we pick out in those blue parental cells shown on the top part of this dimensionality plot on the left, could we pick out the cells that actually have the potential to turn into or to be blue and green metastatic subpopulations? So what you hopefully see here are 2 populations, the blue cells that really mostly cluster separately from the red and green cells. And on the right, we've just kind of made the red and green cells all gray because they're very similar to each other. And so the parental cells are mostly by themselves, but a subset of them you can see do cluster with the metastatic cells. So we wonder if those are those hiding cells that can have the potential to metastasize. And on the right, on the super colorful plot, is really a visualization of all of the 13 clusters that we were able to find in these populations. And I can tell you that of these cells, some of them only look like parental, some only look like metastatic, and some share characteristics of both. And so what we really wanted to do to confirm the heterogeneity of these parental less metastatic populations is to analyze them alone. So for reference, I'm showing you on the top the merged data set that I just showed you on the last slide, and showing you one of our favorite candidate genes from our bulk studies, the SPHK1, which really seemed to mark the more metastatic cells. And you can see that that clusters by single cell seq with the metastatic red and green cells on the left. But as I mentioned, we want to really further dissect just the parental cells to find those needles in the haystack. And that's what I'm showing you on the bottom, the parental only. And within those parental, we do find a subset with that SPHK1 expression, as well as several of the other genes that were from our metastatic signature. And then 2 other quick pearls from this very early data that we have. Next, what we wanted to do really was take advantage of some emerging data and actually studies done by many in the field that's been reported at our recent meetings, as well as published by many groups, and also very close to some of the work done by one of the newest investigators at St. Jude, Adam Durbin. And what we wanted to look at was, we know that neuroblastoma tumors actually have lineage committed but distinct mesenchymal and adrenergic cells. These are genetically identical but develop different phenotypes, essentially due to expression profiles that are defined by these unique different superenhancer transcriptional networks. And the MES cells, or mesenchymal cells, are the ones that sounded a lot to us like our metastatic subpopulations. So we wondered if maybe these were overlapping and shared characteristics. And were these cells we identified in vitro and in vivo, maybe those MES cells that I show on the bottom panel, or again, share similar properties, and that they're not just unique. So we thought we would take advantage of our single cell data to try to get more clarity on whether maybe some of our cells actually are another model of these MES cells. And so this is that same plot I showed you with the blue, green, and red. And on the very right-hand side are looking at the gene sets that have been published that represent the mesenchymal gene expression profile. And they're really those shown on the bottom part, the more pink or red part. I don't know if you can see my arrow, but hopefully you can appreciate the pink and red cells overlap best with the red and green cells. And PRX1 is one of the mesenchymal genes shown in the middle, just as a reference point. So again, we do think there's some overlap probably between the mesenchymal and our metastatic cells, and we're very interested in this and looking at it much more carefully in the coming months. And then finally, the last thing I want to share with you is how we tried to look within these metastatic and mesenchymal populations in our parental cells, again, those needle in the haystack, to look for the most differentially expressed, most highly expressed genes within the parental that also reflect the most highly expressed in those metastatic subpopulations, to try to pick out genes that we could use to tag these or to identify these very metastatic cells within the original cells. And then we predict that might actually then translate to a tumor and be those cells that are hiding. And for a variety of reasons, we picked the gene that I've highlighted on this volcano plot as one of the most differentially expressed in the high, more metastatic cells, TM4 SF1, which I'll just call TM4 for ease on the next few slides. But that is actually one of the genes we saw most highly differentially expressed in our metastatic group, and then found in this separate group of parental cells, but mostly within those that seem to also share other markers of increased spread. So the last few slides just show how the lab, Alex and Alice, wanted to try to characterize whether this was actually functionally true. Does this gene actually mark a population and or have a role in mediating metastatic disease? And one of the reasons we chose this, besides the volcano plot that I showed you, was that this gene has previously been implicated in metastatic outgrowth, and there have been in vitro and in vivo studies showing its depletion reduces lung, bone, and brain metastases in other models of cancer. And so we hoped it might be one of these markers of a mesenchymal or metastatic neuroblastoma subpopulation. And this is very early data. But what you can hopefully appreciate here is if you compare immunostaining of the parental and the bone metastatic group of cells B5, you can see more red surface staining of TM4 on the right side versus on the left side. So we believe that showed there was more protein expression as well at the single cell level on slides. And then what we wanted to next do, which we've now done, is sort those cells. And because this is a surface marker, we were able to do that by flow cytometry. And taking those TM4 positive sorted cells, we were able to show in the diagrams on the left, quantitatively, and on the right in terms of the pictures, that the cells that we selected for higher expression of that surface marker TM4 were more migratory and more invasive in vitro. So what we needed to do next, as I've told you all along, is see if this really happened in vivo, because there are multiple steps to metastasis in vivo, and they don't all involve migration and invasion. So to answer that question, Alex took the cells that highly expressed TM4 and were sorted, and then utilized our metastatic model. And what you can hopefully appreciate in one of the models shown here is that the cells on the top that express this marker TM4 at early stages, and again in very preliminary experiments, seem to show more growth as well as more metastasis. And that's quantified on the bottom. And again, this is super early work, but whether this is a gene that's really a neuroblastoma metastasis gene is unclear. But I think what we have been able to show is at least proof of concept that we can use this approach to identify and select for groups of genes and gene markers that might be populations or subpopulations we want to study, and can use this in cell lines and primary cell lines and tumors ultimately to ask the kinds of questions that I've talked about. And one of those questions, and one of the things we're actively interested in, is whether we can use this to really start dissecting differences even in the expression patterns in different metastatic sites, which up until now we have not been able to do using our bulk microarray studies, looking at the bone versus the brain sites, but hope maybe by using the single cell approach, we'll have better ability to dissect this. And so we really are interested in using this, as I mentioned, this model, not just with this particular cell line, but many other cell lines, primary and established, to identify biomarkers, maybe prognostic markers, although really more importantly, therapeutic targets. And I think increasingly to come up with ways of studying heterogeneity that are ways we can manipulate genes, because a lot of the work to date, which is super important with primary tumors, does not allow you to manipulate things in a system where you can ask questions. And so I hope what I've shown you is that neuroblastoma is really a spectrum, and that's really all about the biology. And I try to tell that to everyone, that it's not just the clinical factors are quite telling, and the presentation of our patients actually tell us a lot, but it's really in the biology. And increasingly, as I show on this slide, I think circulating tumor DNA is going to be another great clue, especially in the relapse setting. But I haven't had time to talk about the importance of thinking of some of these markers and genetics for how we predict late effects, because we are doing better at curing, but the cost for high-risk patients is just too much currently. And there's a great study going on through the Children's Oncology Group right now, led by Lisa Diller and Tara Henderson, trying to better understand the long-term effects of high-risk neuroblastoma therapies, and also with Sharon Diskin at CHOP, trying to understand some of the biomarkers and genomic markers that might help us predict who is most predisposed to those long-term side effects. And finally, I hope that our risk classifiers can move from sort of a few factors and lots of regression tree analyses to maybe thinking of rapid learning and machine learning types of ways of integrating the genomic data and other large-scale data that's going to be coming at us at a pace that's much different from what we've experienced historically, so that we can really learn from and incorporate these lessons to come up with our next risk classifier that I'm really hoping is going to have a lot more biology integrated. And I can't not acknowledge all the great people, and I don't have a great lab picture because of everything that's been going on, but many of the people in the separate photos are more of the recent group that's done some of the studies, and a lot of our work with the KICS team is led from the, led at the therapeutic side in the neuroblastoma group by one of our very talented, very senior fellows, Sarah Kongogo, who's shown here. And then this is our group, our clinical neuroblastoma team, our clinical solid tumor team, who also are critical to all of the work that we do. My family, who I always have to acknowledge for letting me do everything I do, and my husband, who's also a collaborator on some of this work, Michael O. And then the current members, and then the past members who've contributed to what I've shown you. And finally, I can't give a talk without acknowledging all the funds, of course, and the patients who really continue to inspire me, and all my patients, including the one with the star here is especially special to me. It was my first patient who unfortunately passed away, and his mom a few years ago wrote a book about him, which is actually available. And all the proceeds, I think, still go to some neuroblastoma research at Dana-Farber. But it really inspired me to continue on this path, as all these patients do, and all the work that's really important collaborations with the neuroblastoma group and the INRG. So I'd be happy to take any questions. Thanks so much for your attention.
Thanks so much, Meredith, for a really fantastic talk. We do have a number of questions coming in through the chat, and I remind participants to please put those questions into all panelists, and we'll get as many as we can answered in the next few minutes, hoping that many can stay on because Dr. Irwin has agreed for an extended Q&A. But I'm going to jump in with some questions. We've got a bunch in the chat. The first question that came in was actually regarding anti-GD2 therapy and the impact on the number of cycles of anti-GD2 therapy delivered after transplant on their survival of children with high-risk neuroblastoma. Your thoughts, Meredith?
Sure, it's a great question, and I think we're trying in the COG, and when I say we, I mean I'm sort of not the primary person involved, but our group in general, our disease committee, is very interested in this question, and there is a lot of work trying to identify whether the number of cycles, because as we know, sometimes not all patients are able to tolerate the whole regimen, which is currently 5 cycles of anti-GD2 therapy, whether the number of cycles and ability to deliver full doses of cycles impacts overall on survival. I'd say there's some anecdotal and probably very early preliminary data that suggests it might be true, but I think the jury is out. There is a lot of data from all of the work done in the Children's Oncology Group to get GD2 FDA approved. So we do have a lot of that data, and I think it will be important. And I think most of us do feel that the number of cycles is very important. I think the fact that we've been able to take away IL-2, one of the cytokines that we had used for the first more than 10 years of giving this therapy, I think will allow us to deliver more full dosing of therapy because the side effects seem significantly reduced and the efficacy has not appeared to be effective in many studies. So I'm hopeful in the future we'll be able to get more of the GD2 therapy into children.
Great, well, thank you. There's another question regarding combinations of SHP2 inhibitors with other drugs, maybe perhaps even temozolomide. Any thoughts on that?
We've started to look at some of those. We focused initially on, because we didn't get great single agent with the tool compounds, at least, we focused on the pathway itself. But we certainly are looking at sort of the common chemotherapies, the topocyclos, the temozolomide, irinotecan, or single agents, and whether we can do that. There's not a definite mechanistic hypothesis other than we know sometimes we can see synergy with chemotherapies to suggest one agent over another currently.
Excellent. Now a couple of questions about metastasis, and I'm going to bundle a couple of these together. So
Sure.
The first question is, are the metastasis from neuroblastoma osteoblastic or osteoclastic? And then others go on to ask, 2 out of the TM4 SF1 develop metastatic cancer, not all. Is there something due to specific immunity of the animal that could account for this, or other factors you could account for this?
Sure. So maybe the first one, I think I have to say, I don't think there's great work done sort of looking at I'll call it the microenvironment of bone lesions in neuroblastoma. My understanding is we think there would be some osteoclastic activation, but I can't really say that I'm an expert on knowing the sort of ratio and whether there's also osteoblastic activation and how carefully that's done. But now I'm certainly going to look at that a little more carefully. I've always wondered about the osteoblast, just given the neural crest sort of etiology or origin for those cells. But I will get back. Somebody also chatted to me about that, and I can get back to that person as well. In terms of the TM4 and why maybe some of the animals get them, I do think immunity is one thing, and I'll get back to that in a second. I also think we were actually surprised that any genes we looked at, both in our initial paper and in some of the subsequent work, by themselves had that much of an effect, to be honest, because I feel as though it should be multiple pathways and multiple genes that are needed. So I think I'm sure it's not the only one. So I don't pretend to think it's in and of itself TM4SF1 is able to promote this, or that it labels all of the cells. So I suspect there are some TM4 cells that or TM4 negative cells that still retain other qualities that allow them to metastasize. I think what we're interested in doing is look at those that escape to see what's different about them. And then I did want to come back to the point about the immune system. I think a huge weakness of all the models, including the models I talked to you about from our lab, are the fact that they're in immunocompromised mice, and the immune system cannot be studied effectively in not-SCID mice, obviously, which is something many are working on to try to understand better. Obviously, genetically engineered mice get over this because we don't need to do an immune compromised background. But also, I think there's work on humanized models to try to better have an intact immune system to study these things. And we also have a couple early studies. I'm not sure zebrafish will be better than mice per se, but trying to use the zebrafish model as well to get at some of these questions.
Well, terrific. Thanks so much, Meredith. We are currently at the top of the hour. There are more questions in the chat, but we understand that some people may have to drop off. We will get through the questions in the chat since Dr. Irwin is going to stay with us longer. And please, if you can stay, we'd be happy to have you stay and ask additional questions. Also, Meredith, I do want to mention lots of thanks and congratulations for a very nice talk in the chat. And certainly we concur with that. There is a question in the chat that came in about the differences between treating neuroblastoma with other brain tumors like glioblastoma, right, and the brain metastasis pieces that we're beginning to learn about in neuroblastoma.
Yeah, I mean, I think certainly the genome of glioblastoma is a bit more complicated than neuroblastoma. So when it comes to specific pathways and specific genes, whereas they obviously both have the same issues with getting across the blood-brain barrier, but I think it's hard for me to imagine other than that there's that much as a group that glioblastoma and neuroblastoma have in common. I could imagine there could be pathways and subsets of each that may be shared, just like neuroblastoma shares, ALK aberrations, although different ones, with lung cancer. But I'm not sure it's the same issue completely. I think one of the other differences in neuroblastoma is we mostly seem to see metastases to the brain sort of towards the end of a patient's sort of chronic journey, or maybe brought out by the use of immunotherapy potentially. So I think the factors that sort of contribute to it starting are quite different, but the blood-brain barrier is for sure a shared issue with both. So we're definitely, in fact, some of the labs we collaborate work on glioblastoma because they obviously have the interest in compounds and drugs that can cross the blood-brain barrier. And a similar challenge we're all going to have, I didn't talk at all about CAR T. There's obviously lots of interest at St. Jude and elsewhere in using CAR T for solid tumors, including brain tumors. And so if we do want to ultimately affect CNS recurrences, hopefully we can learn from our colleagues trying to use this strategy in the primary CNS tumors.
Terrific. And Meredith, you mentioned, as you developed your model to make it recapitulate what we were seeing in patients, that you were able to have the neuroblastoma cells honed to the adrenal gland. And the question that's in the chat is, why does that? Do you understand why that happens, or why that's a common site of primary disease in patients?
Yeah, I don't think we know. I mean, what's interesting, I didn't have time to show this in detail, but the adrenal, so-called adrenal metastases actually cluster more with the parental cells, whereas the ones that go to other parts of the mouse body, to the bone and the brain, cluster separately, at least from the bulk sequencing and the bulk expression work that we've done. So they don't seem to be, like when we re-inject those cells, they don't seem to be as aggressive as the others. So I'm just presuming there's something about that microenvironment that neuroblastoma likes. I should say that even though I said it was the adrenal, there's also stuff in the sympathetic chain there that in the bioluminescence, it's hard to distinguish if we haven't opened up the mouse or done a three-dimensional imaging with CT or MRI.
Great. The next question is in relationship to ALK and ALK. Have you identified ALK dependence or ALK independent mechanisms in neuroblastoma patients treated with lorlatinib?
So for the drug, I guess, is what the question is. So, I mean, I think unlike some of the kinase inhibitors many of us use now in other solid tumors that are more promiscuous kinase inhibitors, I think it's felt that lorlatinib, especially, and more than the earlier generation ALK inhibitors, is a bit more selective for ALK. That being said, I would never say that it can't work against other either other kinases or potentially other other pathways that we just don't understand. I think what probably more of the research is focused on are the escape or resistance mechanisms when we use drugs like lorlatinib. But I would not be surprised if, it'd be interesting to take a cell where I guess you take out ALK altogether and see if lorlatinib has an effect. My understanding is in wild type ALK, there's no effect. And we've seen that in the lab too when we use any ALK inhibitor. But whether that's maybe, not the case for a small subset, maybe there's some other drivers or other things happening. Neuroblastoma is so heterogeneous, I would imagine there could be a patient somewhere that responds to an ALK inhibitor without a clear mutation.
Well, thank you. And the questions keep coming in, and we encourage everyone to keep putting those into the chat. The next question is with respect to your comments about clonal evolution, and it is regarding MYC-N status. Will chemotherapy cycles alter the MYC-N status in patients?
That's a great question. We wondered about that, and actually, I don't know, almost 10 years ago now, we did a much less mechanistic kind of study with one of our pathologists, Paul Thorner, who's since retired, but used to work with the POG neuroblastoma group. And he actually wanted to just look at MYC staining as we treated patients. So essentially their initial tumor samples and then their post-chemotherapy resections. And we did find that often we did not see as much MYC expression. At least this was by immunohistochemistry and immunocytohistology and various other techniques. I think it's not clear. Certainly, we always say that once a MYC-N amplified, always a MYC-N amplified, but I'd say most of that data is pre-NGS, where we can look much deeper and much more at heterogeneous populations. My own thoughts from having sequenced a few different bone marrow samples too from patients with MYC-N amplification is that it's heterogeneous. We actually know that from the FISH that we do on the primary tumors at diagnosis. And, I've had at least one case where we've detected MYC-N amplification on one side and not the other. So I do think it's heterogeneous, and I'm sure we're selecting one way or the other. I think we're selecting, at least initially, if patients are responding for the non-amplified cells, but who knows if there's a few of these hiding out, and those are likely, because usually the MYC-Ns, when they come back, if we check again, they're still MYC-N amplified. I don't know if that helps. It's kind of hand waving, but
No, it's always a good question, right? And, clinically, what we thought we once knew may be evolving, and I think that's part of why taking care of patients with neuroblastoma is something that many of us as pediatric oncologists are always intrigued and inspired by our patients. The next question is along those same lines: is what type of mechanism does neuroblastoma follow vis-a-vis the clonal evolution or cancer hypothesis theory?
I don't think we really know. I think there are just definitely, I don't know if it even fits either, to be honest. The clinical things I see, and I'm sure Beth and others do, is you can have a patient with diffuse metastatic disease and a huge tumor, and sometimes the huge tumor responds and the Mets don't respond, and sometimes vice versa. Sometimes some Mets respond and some come back, or the so-called mixed response, we call it. So I really do think there's clonal heterogeneity, and I do think over time there's evolution, but I think we drive that with our therapies to a certain extent. I do think circulating tumor DNA is going to be so interesting, because what it'll hopefully tell us is that maybe we don't have to keep biopsying patients, at least to see dominant mutations. We're not going to see everything, probably. But, even though we say clonal evolution, I've just had a patient that had a 40% variant allele frequency of a clonal ALK mutation, and 5 years later recurred in the same place. It's 38% variant allele frequency. So some recur, and are completely different, and sometimes it seems like nothing changes. But as Beth said, every patient seems just a little different. And I haven't even thought of the germline yet, which I also think plays a huge role in these neuroblastoma patients as well.
Great. Well, thank you. And the questions just keep coming in, and they're all great ones. So with respect, you spoke about amplification of MYC-N gene. Are there other things about MYC-N? Have you seen any examples of downregulation of MYC-N? And you mentioned some of the other genes that gene expression that may be downregulated. Any additional comments there?
I think, certainly MICN does a lot of different things. And I would say some of the really cool super enhancer work done by Adam Durbin and others, I think will really give us more of a important insight into what MICN can do kind of after the genome's been determined in terms of the expression, this sort of post-genetic lineage kind of commitment stuff that we heard about. So I do think MIC does a lot of other things. And I'm sure some of it is downregulating things, if that's what the question is, as well. And I didn't even mention, we're talking about MIC-N, but there's also quite a lot of data to show that in a subset of patients that don't have MIC-N amplification, c-MYC is probably a driver. So much like the medulloblastoma story, where that's probably the opposite. There's a bit more c-MYC patients and rare N-MYC. Here it's probably the opposite, but I do think MYC itself is probably quite important beyond just MIC-N.
Great. So more questions about metastatic spread. And, do you think, is there any evidence from your lab or your thoughts on the metastasis that go to lymph nodes? Are they biologically different than what goes to bone, and, the types of lymph nodes they go to, and the type of lymph node spread that you see?
Yeah, so I think so for sure. So that was another example of, there were these patients in the clinic that the neuroblastoma only spreads to lymph nodes. We used to call them stage 4N for nodes. And there was actually a small case series from Dana-Farber, but no sort of large studies. So with the power of the INRG database, which is now over 20,000 cases, we were able, this is work led by Daniel Morgenstern when he was a fellow, to look specifically at patients with lymph node only disease and show, as we all knew from the clinic, that they do have a better prognosis. We have wanted to sequence and especially look at expression profiles in those patients to see if there's something different, because we do know that liver patients do horrible other than the little babies. Lymph node patients do really well, and bone marrow are sort of closer to the liver, farther from the lymph node. So we haven't been able to do those expression analyses. Other cancers, they have done it, like melanoma breast. There is a study, I believe it's from at least 5 years ago, from the COG in rhabdomyosarcoma, looking at expression profiles of different sites, the nodes versus other sites, and seeing differences. I think those would have been microarrays at that point, but different expression patterns. I'm sure the expression patterns are different, probably due to the microenvironment niche kind of requirements, for sure. We just don't know what they are yet.
Fantastic. There's more thanks and appreciation for the great lecture in the chat, Meredith. I'm going to let you take a break for 1 second and see if there are any others that come up in the chat, and just have a public service announcement for the next Science of Childhood Cancer presented by Olivier Delattre, and he will be talking on I'm looking for the title in the chat here, and I may
Ewing sarcoma, probably.
Yes, absolutely. How did you guess, Meredith?
Oh,
and
yeah.
We look forward to having folks join for that as well. I don't see any other questions coming through in the chat, Meredith, and we appreciate so much your being able to stay over. I guess I would ask one question, Meredith, as you sort of we wrap up. One of the things I didn't bring out in your introduction was the tremendous number and success of people that you have mentored. And so if you could give us any thoughts on both how to be a good mentor and how to be a good mentee, I think we would all benefit from that.
Oh, wow. I wasn't thinking of that on my list of questions that people would ask today, but it's something I'm very passionate. It's probably why I do some of the jobs that I do. I think, I guess I would say in terms of sort of research, whether it's clinical, which I also do, or basic research in the lab, I think is really finding a mentor who shares your passion for questions, and also cares hopefully about your career development, and that you're doing something that's going to get you to the next level. And so I try to think with especially with clinician scientist trainees, but I also have basic trainees as well, and ones who don't really want to do the basic work, but what is sort of their their goal overall, the types of questions they want to answer, and how can they get there, and try to find a project that works. And then for me, especially as I've become increasingly busy with other things, is making sure what people want to do aligns with what I see my lab and my clinical program doing, so that I don't take on things that are too far away, because then it doesn't work out. But I would say, as a mentee, not to be afraid to ask mentors to be a mentor for you, and that you always should have more than one mentor and more than one advisor. You know, somebody who maybe helps you with your career may not be the same person who helps you with a research project. I think those are conflicting, and may not, there may be unusual, amazing people who can do both, but it's pretty hard to mentor someone's project and mentor their career at the exact same time, to get the right outcome. But never be afraid to ask. I think, mentees always want mentors to call them, and, mentors might be busy. It doesn't mean they're not thinking of their mentee. It just may mean the olive branch, at least after the first meeting or two, may need to come from the mentee. So I don't know if that's helpful or not, but I love to see my trainees kind of go on to start their own labs and start their own programs in other countries, in Canada, in the US, wherever. It's like having kids. They're different than my kids who will never do science, but it's nice to see them doing things that they're passionate about.
Well, again, thank you so much, Meredith. This was a fantastic presentation. Thanks for answering all the questions and really leading us to think about neuroblastoma and where we can go in the future. And, for sharing your ideas on mentorship and your passion for, treating patients, doing work in the lab, and really making a difference for the outcome of children with neuroblastoma. So thanks again, Meredith.
Thank you. Thanks, Beth, for the invitation. And I hope to see people in real life soon. Okay. Bye.
Bye-bye.